Yes, good day, class. I welcome you to today's segment, Life Science Grade 12. Last week, I've been dealing and presenting to you DNA structure. Today's presentation, we are going to be looking at the difference between RNA and DNA. So I think I've said a lot and I've given a good presentation on the two, actually on DNA. So on the board here, you can see the structure of RNA and the structure of DNA. And below it, there is you can see below the tab, below the structures, there is also a table that is going to be used in analysis of the two nucleic acids, the DNA and the RNA. So please take note that actually I want to start by bringing to you the RNA, and then we come back to this presentation. So I'm just giving you to see, or I'm displaying on board right now for you so that you can see what I'm going to be presenting and talking about today. So therefore, everyone can just keep logged on and understand these two molecules by structural appearances. So let us just go to the beginning, or let's just start from where we are supposed to start on, and then the differences between DNA and RNA. I don't want to talk anything about DNA. Like I said, DNA, I've dwelt a lot on it. So what is RNA? It is also a type of a nucleic acid which is directly involved in protein synthesis. I said last time that DNA, it actually has a genetic code that is coded or that is transcribed to RNA, whereby RNA actually mRNA is formed from DNA. So what we need to understand is that uh, all the information that is in the DNA molecule, it is also transcribed, or it is synthesized into what into the RNA structure. So one key thing is that we have got two types of nucleic acids, which is RNA and DNA. So RNA, it is actually the one that is directly involved in protein synthesis, which is translation. Protein synthesis or translation. I'm sure last time I talked about that. Protein synthesis or translation, the process by which proteins are formed. So it is also known that RNA is an abbreviation that stands for ribonucleic acid, which is an important nucleotide which with long chains of what with nucleic acid present in all living things anyway. Living things have also have got nucleotides, or it has also the RNA nuclear it has also the RNA structure, which is also responsible for what? Nucleic acid, that is also responsible for protein synthesis. I'm sure that you remember and you understand that cell division has to take place, cell growth has to occur. The growth of cells, the replacement of dead cells, the elongation of cells, it is happening as, or it has to take place as a result of the uh, functioning of the RNA nucleic acid. So its main role is to act as a messenger. RNA, its main role is to act as a messenger, conveying instructions from DNA for controlling protein synthesis. Instructions class from DNA that controls protein synthesis. I'm sure that you understand that the DNA, we say that it controls the structure and functioning of an organism. So in that case, what you need to see is that there is a direct relationship, which is, you can say it's a biological relationship, which whereby the RNA, the mRNA, it has to encode in it the information that it assimilates from the DNA, such that it can also be sending that signal that must be sent, or that must be contained the genetic code that is being sent, or that is assimilated from what? From DNA, in order to allow, or in order to promote the formation or the synthesis of proteins. So RNA contains ribose sugar, it does contain ribose sugar that I'm sure you know. Last time I understand I did mention that again, DNA, it contains deoxyribose. So RNA, it contains ribose sugar. It also, of course, you have the phosphate. It has also have the nitrogen bases. Likewise, there are four nitrogen bases that are there in what in RNA. We also have the guanine. We have got the adenine, we have the guanine, we have the cytosine, and again, I'm saying we have got the outstanding or the unique one, which is the uracil. Uracil, I'm sure that you remember even last time when we were presenting on DNA, I mentioned to you that uracil is a unique nitrogen base that is oftenly found in what? In RNA alone. So instead of thymine, we have got uracil, as you can see there. I'm sure you are going to see again as I go down with the presentation. So it contains uracil RNA instead of thymine. DNA and RNA share nitrogen bases. Like I said, there are three nitrogen bases that they share. This can also appear in your multiple choice question to say identify the basic or the three nitrogen bases that are common in both RNA and DNA nucleic acids. So what is important to, or what you must know is that the two of them, they both contain, they both have, adenine, they both have guanine, and as well as what as cytosine. So thymine is usually only found in DNA. Thymine can only be found in what? Thymine can only be found in what? In DNA class. Only take note of that. It is only present in what? In DNA. Thymine is only present in what? In DNA. That I'm sure you should understand. And uracil is also usually only present in what? In RNA. So what you should see is that there is a distinctive difference there, or a structural physical difference, which is to say that uracil is found in RNA and DNA, it has got thymine. So without wasting any time, that is just the introduction to RNA. Let's go to types of RNA, which I should understand and you should know as well that only some of the genes in the cell are exposed into what? Into RNA. But we also have some types of RNA wherein each type is encoded by its own type of a gene. So what you need to understand is the types of RNA that we have. We have got tRNA, which I'm sure you know that is called what? It is called transfer RNA, which you can see written in short as it is in this case, tRNA, transfer RNA. Transfer RNA, for those who may have gotten a chance to read IAT or they've seen it in previous papers, you can see that there are two main types of RNA. Actually, there are three of them, which are mostly dominant. So the transfer RNA, which we say is tRNA, is a small molecule in cells. It is small, that carries amino acids to organelles called the ribosome. It carries to the ribosomes where they are linked into proteins. So tRNA plays a crucial role in the process of translating genetic information into proteins. So as we are going to go into the next presentation, what you should perceive right now is that tRNA, it it brings amino acids to the ribosomes, which I'm sure that you remember the process of complementary pairing of the codons anticodons relationship. So the tRNA plays a very important role as it brings the corresponding amino acids that must also be used in the translation of genetic information into proteins. So that is tRNA. We have got mRNA, which I am saying again, it is in full, it is called messenger RNA. Messenger RNA, it is that type or is that type of RNA that is formed during transcription. So it's a single-stranded molecule of RNA that correspond to the genetic sequence of a gene. It is a single strand. Anyway, RNA is naturally a single strand, which we are saying it corresponds to the genetic sequence. It corresponds to the genetic sequence of a gene and is read by the ribosome in the process of what? Of synthesizing proteins. I just want to show you a simple, what you call diagram, which can relate to what I'm saying right now. mRNA is a single strand which has to be read by what, or which must go through, or which must go under the what, the ribosomes in order to allow the ribosome to send the relevant signal on which form of amino acid must be brought back by tRNA. So this is our mRNA, which is a single strand. It's our mRNA there. So what happens there, we are saying here is the what? Is the ribosome. So what happens is that on the ribosome, it is a site, it is an industrial site where proteins are manufactured. So what we are saying here now is that the mRNA, it has to go through this point. It goes through this point or underneath, or it goes through the industrial site, which I'm demonstrating here. So as it passes, it is passing in this direction. It is passing in this direction such that it can cause what? It can be read by, or it can be decoded by this. So as it passes here, we are saying the codons which are found on the mRNA are read one by one in order to relate to the mRNA, to the transfer RNA. So therefore, a process of protein synthesis can take place. So mRNA is created during the process of transcription. I'm sure that I've mentioned already that it is produced during the process of what? Transcription, where, or it is through the help, or the catalyzation, which is brought up by the enzyme what? RNA polymerase that converts the gene into a single-stranded ribonucleic acid product synthesized by transcription of DNA. So it is produced during transcription because of the DNA polymerase enzyme that catalyzes, or that activates the process of what? Of transcription. So therefore, it is actually produced as a product of what? Of it is a product of transcription, that one. So genetic information in mRNA is contained in the sequence of nucleotides. The genetic information in mRNA, it is contained in the sequence of nucleotides. I'm sure that you know nucleotides are the building blocks of what? Of nucleic acids. I can give to say that nucleotides are building they are building blocks of nucleic acids. When you're talking about nucleic acids, I'm sure you know that I mentioned to you that it is what are the building blocks of what? Of nucleic acids, the nucleotides. Nucleotides class are the building blocks of what? Of nucleic acids, which we are saying the RNA and the DNA. So which are arranged into codons containing of three ribonucleotides each. So in this case, we are talking about the nucleotides that are specifically in what? In the which are specifically in mRNA. So each codon codes for a specific amino acid. This must be known. Each codon code for a specific amino acid. We have got plenty of amino acids, which we say that they are the building blocks of proteins. So what you need to understand in this case is that each codon codes for a specific amino acid. I'm going to go into the details of the codon-anticodon relationship as we go to the processes there. mRNA is the blueprint for what? For polypeptide synthesis. So as to say it's the blueprint, we are saying it is the guideline, it is the framework from which polypeptides are produced. This is just a long chain of what? Of amino acids. To say the long chains of amino acids are produced or are manufactured as a result of what? mRNA synthesis or mRNA being read by the what by the ribosome. So please understand that it is actually the one that gives a specific order in which proteins are synthesized. So therefore, we are talking about the polypeptide synthesis, just to say there are multiple chains of amino acids that are produced. These are a product, or they are produced as a result of what? Of how the mRNA is being uh, worked upon by the ribosome. Uh, let's go to the next type of RNA, which is number three. We have got number three. Number three, the ribosomal RNA, which is called the RNA. It is actually mRNA. It produces ribosomes with the ribosomal proteins that are organelles responsible for translation of what? Of the mRNA. I'm saying organelles that are responsible for the translation of mRNA. It actually provides a structure for the workbench where polypeptides are assembled. So what I've been saying, or what I've been trying to illustrate above there, is that it actually provides a workbench, as to say a work site, or it is an industrial site. In other words, the workbench, as to say an industrial site where polypeptides are assembled. So in terms of this, the chain that is produced after mRNA, or after the codons have passed through the ribosome, we are saying it is actually worked on because of the ribosomal RNA, which assembles these polypeptide, uh, the polypeptide chain or bonds. So the incorporation of amino acids in the exact sequence requires the correct binding of specific what? The incorporation, or the bringing together of amino acids in the exact sequence, it requires correct binding of specific transfer RNA to mRNA guided by codon-anticodon pairing. I'm sure that we talked about that, to say that there is a single strand of mRNA that has to match its codons with the anticodons that are brought by the transfer RNA, which is having some specific amino acids, which are therefore synthesized and producing a specific chain of what that has to be produced in that regard. So what I'm saying is that these are the three types of RNA. So in summary of types of RNA, in types of RNA, we have got, we can give to say that we have got three of them, which I can easily identify to you right now, to say that we have got what? We have got three types of them, which is the tRNA, we have got the what? The mRNA, and then finally we have the what? We have the RNA. So these three are the major, or these are the three main types of the RNA that we are expected to know of. So please take note of that, and I'm sure that you are not gonna make mistakes. You are not gonna make mistakes on this, and you get them right as you go. Because in the next presentation, you shall see that we are going to be talking about their functioning, or how they are influencing various processes. So understand and get it right that these are the three types of what? Of RNA that we have. So we can go to the differences between RNA and DNA. I just drew some diagrams here. I've drawn, or I've captured nice diagrams that you can see here. So we are having our what? Our RNA here, ribonucleic acid, which is given here, which I can give to say one on this side, and then we have got our DNA, which is deoxyribonucleic acid, that we can also write to say is two there. Can we agree and see that. So you can see that we are saying the structure. One thing that we need to understand is that RNA, the RNA structure is a single strand that you are seeing there. You are seeing that this is a single strand with its nuclear bases, which we are saying these are the bases that are there. Like I mentioned in the first place, that we have got what? We have got cytosine, we have got guanine, we have got adenine, and we have got uracil. So these are the three, the four nitrogen bases that are found on the single strand. It doesn't have a double helix structure. And then we have got we have got versus here as to say compared to what? Compared, compared to what? To DNA. Compared to DNA. So what you can see here is that this is a helix of phosphate sugar phosphate, a helix of phosphate sugar sugar phosphates, which I say that the strands of nucleic acids are formed because of, or they are formed from what? From phosphate sugar molecules. They form these strands, whether it is in DNA or it is in RNA. The strands, which we say the legs, which we can sometimes define as the ladder-like structures, are the legs of the ladder-like structure are phosphate sugar molecules that forms them. And then on the same structure here, on another way, on the other side here, which I said too, we have got our cytosine, which is also given here. We have got our guanine, we have got our adenine, we have got our thymine. And these four also constitute the nitrogen bases that are found on DNA structure. So you see, we have got strand 1. You can see that we have got strand 1 there. We have got our strand, I can say strand A, and then we have got strand B, which are both found on what? They are both coexisting, or they are both found on what? On the DNA structure. So you can see in this regard that we have got our G, we have got our C, and then we have got the weak hydrogen bonds that are existing in between there. The same applies to all the other various nitrogen bases that are found up to the end there, where we have got our thymine and our adenine that are also indicated on the structure, as you can see there, our adenine, and then our thymine and adenine bases that you can see there, the thymine adenine relationship, the thymine adenine relationship that you can see there. So let me just go to the structure and explain it clearly according to the notes that I have put together here. So DNA, deoxyribonucleic acid, the RNA, which is also the ribonucleic acid. By definition, you can see that it is a long polymer. DNA, it is long, and then it has a deoxyribose. It has a deoxyribose and phosphate backbone having four distinct bases. There are four distinct bases. I already mentioned that, and that can actually be understood very well, that there are four distinctive bases which are mentioned: thymine, adenine, cytosine, and guanine. Then in RNA, it is also with the ribose and phosphate backbone with four varying bases again, which is our uracil. We have got our cytosine, we have got our adenine, and we have got our guanine. So those are the four nitrogen bases that are found in what? In RNA. I don't want to overemphasize because we have already talked a lot on that. Let's look into the location. Location, in terms of the location here, in terms of the location, what we are saying, or what you need to know, is that as you can see, these two nucleic acids, understand that where are they found and how are they influencing their processes. So as you can see, I'm saying we still have our DNA, and we have got our RNA. So DNA is located in the nucleus of a cell and also in the mitochondria. This we are talking in regard to, or it is expressed in regard to animals. So it is located, it is found in the what? In the nucleus. It is found in the nucleus. It is also found in the what? In the mitochondria. So DNA, in terms of its location, I have also mentioned in passive in the previous presentation that DNA is found in the what? In the nucleus of a cell and then in the mitochondria, which of course it is in animals, which at another time you might be asked in plants. That is to say that in plants it is found in the chloroplast. So RNA is found in the cytoplasm, it is found in the nucleus and also uh, in the ribosomes. So in the ribosome. So what you can see is that there are different locations here. So in this case, just to make it clear to you guys, is that when we are talking about the genetic information, the inheritance and heredity, we refer to the DNA that is found in the nucleus of a cell, which is most importantly in the what? In the gametes, the sex cells, the sperm cells, and the what? In the egg cells. Of course, I remember you are going to look at mitochondrial DNA, which is involved or it is functional for purposes such as what? Uh, uh, Ancestry relationship, tracing of ancestry relationship, which is actually one other thing that we can look at and discuss as we go to evolution. But anyway, for now, just know that it is found in the mitochondria, and again, it is only found in two places, nucleus and mitochondria. And then the sugar portion, DNA has got deoxyribose sugar, DNA, it has got deoxyribose sugar, and RNA, it has got the ribose sugar is one, one other structural, one other difference that we need to take note of. Function The function of DNA is the transmission of genetic information. I'm quite aware, I explained that genetic information in organisms is inherited. It is passed to generations because of DNA. So it acts as a medium for long-term storage of what? Of the characteristics of organisms. So it acts as a medium for long-term storage. And then we have got the RNA, which is critical for transmission of the genetic code that is necessary for protein creation. So remember that the genetic code that is in this regard, it is specifically important for for protein synthesis, which is from the nucleus to the what? To the ribosome again. So it is specifically important for protein synthesis, such that you understand that RNA, it is highly important to influence and ensure that dead cells are replaced, worn out cells are replaced, and also cell growth. So what I'm saying is that the genetic information, the genetic code is supposed to be what? Is supposed to be coded into what? Into the RNA in order to ensure that an organism remains the same. There is no distortion of characteristics or anything. So therefore, there is a continuity or perpetual existence of organisms. Let's look into the predominant structure. DNA is double-stranded molecule that has a long chain of nucleotides. It has got it has a long chain of nucleotides, and then that is about double-stranded thing again in terms of the DNA. And then RNA is a single-stranded molecule which has a shorter chain of nucleotides. That is also a specific difference that you can find and you can look at. Propagation, we are talking about the what? The formation of it or the spreading of it, the propagation of either DNA and RNA. So what we need to look at again and understand is that DNA replicates on its own. I have got the previous presentation which I explained about the replication, DNA making a self a self, I mean, it makes a copy that is alike to itself, identical copy of its own. We call that replication. So it replicates on its own. It is self-replicating in DNA. It makes it reproduces itself. So RNA does not replicate on its own. It is synthesized from DNA when required. For clarity, I'm saying it is synthesized from DNA when required during transcription. During transcription, it is required to allow protein synthesis. So whenever proteins are to be produced, we are saying a structure of mRNA must be formed. So RNA does not replicate on its own. It is synthesized from DNA whenever it is required. And then nitrogenous bases and pairing. I've already mentioned again, but for just repetition and clarity again, the base pairing, we say that it is what GC in DNA, which pairs G and C. We are saying guanine pairs with cytosine, and also we have got what? We have got AT, which is adenine pairs with what? With thymine. I'm sure that I already told you to say equal sign, it means pairs to, or we also have C pairing with what? With the G. This is the complementary pairing, of bases in what? Complementary pairing of bases in DNA. So classic, please take note of that, understand this, and don't make mistakes. I'm sure that this is quite clear, and I'm trying to write I've tried to type the notes and make them visible and clearly readable, that you can not make mistakes as it comes to the various explanations. So you can see that the base pairing is as follows. We are talking, you need to see again that they have got guanine, cytosine, adenine pairs with uracil, and understand that this pairing, it is only during specific processes. This pairing class only during specific scientific processes, i.e. transcription, translation. e.g., we are saying i.e. transcription or translation processes. So the pairing, understand that in its normal biological structure, RNA, it has got no pairings, but it only does pairing when we are going to look in the processes that are supposed to take place where they are DNA structure has to pair with the RNA in order to produce or in order to facilitate some specific processes, as I've indicated here. So thank you very much for your time, and I'm sure that you look forward to the next presentation. We are going to talk about transcription, translation. These are going to be well explained with their specific diagrams, and understand that each of the processes that we are talking about in this first chapter, transcription, translation, as well as DNA replication, you have got their diagrams that are used in the processes or in the explanation of the processes. Take note of those diagrams. Don't confuse yourself or don't confuse them, but make sure. Thank you very much for your time, and you have a beautiful day. Keep connected. Keep turn on your notification button, share and like the videos so that we can enjoy the channel. You have a beautiful day. Thank you.